The overall goal of this procedure is to identify transcriptional regulators of any gene of interest using a simple, high-throughput transfection protocol and a homemade dual-glow luciferase assay. This is accomplished by first designing and cloning a unique set of dual luciferase reporter plasmids. These plasmids are co-transfected into 293 T cells in microplates with plasmids from a DNA expression library, such that each well is transfected with a single library plasmid. After 48 hours, Firefly luciferase activity is assayed, followed by an assay of Renilla luciferase activity. The final step is to calculate Firefly to Renilla luciferase activity ratios. Ultimately, the relative induction of the gene of interest in response to each library gene is inferred by the ratio of Firefly to Renilla luciferase activity in each well after plate-based normalization.
The main advantage of this technique over existing reporter assay protocols is its ability to rapidly generate functional data for a large number of genes with minimal manpower and cost.
One day prior to transfection, designated as day 1 of the screen, 293 T cells are seeded into 96 well plates. Cells are trypsinized, counted, and resuspended to a concentration of 1.5 times 10 to the 5th cells per milliliter in DMEM containing 14% FBS. Pour the cells into